Hello everyone, now welcome to part 2, amino acids and protein structure. First of all, what are proteins? Proteins are linear biological copolymers of alpha amino acids, which are shortened as AA amino acids, linked through amide bonds between carboxylic acids of one amino acid with the alpha amino group of a second amino acid. Well, let me summarize this as this. They are copolymers, and we have a link between two amino acids. They are formed of many amino acids, by the way, and they are formed between many amino acids where they are linked through amide bonding. And now we are going to discuss all of this. Living things are composed of what? Living things composition, let's say. They are composed of three different materials. First of all, they are composed of four different materials, but we will first focus on the first three, which are the carbohydrates. And then the proteins. We have the fats, which are also referred as lipids, and also we have the nucleic acids, which are RNA and DNA. Alright, now carbohydrates are actually polysaccharides, right? Polysaccharides come together to form carbohydrates, and polysaccharides are of course composed of monosaccharides. And now I will show you the structure of these in a minute, but also let me say you that these are the polymers. These two are the polymer, and polymers are composed of the monomer structures, which can be composed of three different material, which is first glucose, or fructose, or galactose. Alright, and these are the monomers. Now let me show you the structure of glucose, and we have a ring like this, and we have the hydroxide bonding over here. And in here we have another bond, which is, now let me show it like this. This is it. This is our glucose. And of course, when we put them together, we are going to obtain our polymer. So let me show you the structure of carbohydrate, which is just the combined version of this one, which we have the exact same molecule. Let me write the exact same molecule over here, but instead of this group, I will just write CH2OH, so that it will be easier and faster for us to go on. And now we have our molecule, and I will write the hydroxides as before. and we have hydroxide here. We have CH2OH, and here we have another same structure. Let me just copy and paste it over here. And what is the solely difference is that we take these two and we will take a water out of it. So when a water comes out of here, we will just have oxygen, which will be remaining over here. We will just have oxygen connecting these two molecules. Similarly, we won't have hydrogen over here. We will just have oxygen, and we will have our usual molecule like this. It is exactly the same. Alright, and now let's move on to proteins. Proteins are made up of polypeptides. And polypeptides are made of amino acids. Amino acids. Alright, now the amino acid is basically the structure of amino acid can be shown such as this. We have a carbon, which is called the alpha carbon. And we have an amino group attached to it, an R group attached to it, and a carboxylic acid. And we have one hydrogen. Amino group and carboxyl group we have. And this is the general structure of an amino acid. Now, let's continue with lipids. They are made of triglycerides. Now, triglycerides are made of glycerol plus free fatty acid. All right, so what is this about? Now, I will draw you a free fatty acid first. Now we have a long chain, and we have the carboxylic acid at the end, and we have it is called triglyceride, so we have three free fatty acids as it is in the name. And here we have, and this is the part which is called free fatty acids. This part. And oxygen is not included. This is free fatty acids. And I will draw you the glycerol parts, and we have the carbons in here, and these carbons are all connected to the oxygen near them. And these are also connected to themselves between each other, and we have the hydrogens as usual. And this is called glycerol. Well, as a result, the final structure here, this one, let me show it like this to make it more explicit. This structure over here is called triglycerides. Okay, and that's it. And now I will make you an exception. Now, as you know, let's write a quick remark over here. Our remark says that the amino acids have the amino group and carboxyl group attached to the same carbon atom, which is called as the alpha carbon. So we have the carbon atom, and this is called the alpha carbon, and we have the amino acids have the same amino group. We have the amino group over here, and then we have the carboxyl group, and then we have the R, we have the H over here, and this is the alpha carbon, and as we can see, this is always the case. However, we have one exception, which is gamma aminobutyric acid, which can be abbreviated as GABA. Now, why is this an exception? Let me write it here, the name of this, gamma aminobutyric acid, right? Which is abbreviated as GABA, because we have the G over here, A over here, B, and A over here again, which is abbreviated as GABA. Now, as I said here, we have the alpha carbon over here, but however, in this exception, we have the gamma carbon. The gamma carbon is the third carbon away from the carbonyl one. This is used as a neurotransmitter inhibitory neurotransmitter. And this one, the NH2, which is the amino group as we know, is bound to the gamma carbon. To gamma carbon. Alright, great. 3 carbons away from the carbonyl one. What is gamma carbon? It is 3 carbons away from the carbonyl one. Let me write it over here. 3 carbon away from carbonyl one. Alright, that's great. Now let us check the amino acids. Alright, amino acids can be analyzed in 2 groups. First group is called proteogenic amino acid. Proteogenic amino acid, and the second group is called as non-proteogenic amino acids. Alright, so what's the difference? The proteogenic amino acids are actually coded by a genetic codon. Coded by genetic codon, and it is used to code a protein as it is seen in the name. It's incorporated into proteins. Nevertheless, this one is not coded by a genetic codon. Not coded by a genetic codon. Alright, and it's not incorporated into proteins. And I want to give general rule where in humans, in eukaryotes, the monomers have the D structure, and the amino acids have the L structure. However, cysteine has R structure. The reason why is because the R group is different. Now, in order to say why the R group is different, we actually need to know all the amino acids and their structure, and we need to analyze their R groups differently so that we can see which one of them are aliphatic amino acids, which ones are acidic, which ones are basic, etc., etc. Now we will continue these in next lesson. Thank you for listening. Have a